Imagine a beautiful place in a pine forest between two huge rivers. It's cold outside, about 5 degrees Celsius, and we see a cave in the distance. We can also see a big bear and a couple of deer hanging out by the river. And who else? There's a small group of people, perhaps a couple and their children. They're sitting by a fire roasting an animal. The woman seems to be preparing a piece of meat. Meanwhile, the children are sitting at a distance making stone tools for their parents. Let's think about this very detailed picture. Is this what a random day in the Paleolithic was like? And how do we know this? There are many different archaeological disciplines that work very hard to approach these remote time periods. Each of these disciplines contributes a little bit to the big puzzle of prehistory. Archaeological clues can be found in bones, stones, sediment, and other materials with high preservation potential. And it is the results of studying these materials that eventually become pieces of the big puzzle of prehistory. So why don't we take a closer look at one of these pieces? What are lipids? Lipids are essential organic compounds such as fats, oils, and waxes. Since the origin of life, living organisms contain lipids. They need them for strength and protection. Nowadays, we can find ancient lipids that can tell us stories from the deep past. This is because lipids are hydrophobic. They do not degrade with water. That means that they preserve well through time, even up to millions of years, given the right conditions. So what are lipid biomarkers? Lipid biomarkers are the fingerprints that lead us to lipids of specific plants or animals and to their metabolic processes. Thanks to these fingerprints, it is possible to know which plants and animals lived in that place when we find a lipid residue. So what information can we obtain from this technique? Lipid biomarker analysis. For a long time, archaeologists believed that finding organic matter in ancient sites was a lost cause. Wood, clothes, food, and all of the organic human world degrades in years or decades. It's gone. And this leaves us with the poor, very fragmented human story, because most of the human world is organic. But now we know that archaeological materials and sediment can very well preserve DNA, proteins, and lipids, all organic residues. All 3 types of organic molecules are a hotspot of archaeological research in the last 10 years. Different techniques to analyze and to identify DNA, proteins, and lipids are thriving in the archaeological world. In fact, there's a name for this. It's biomolecular archaeology. Lipids in particular, they can approach us to different kinds of plants and animals that were used or consumed, and they can also inform us about climate and vegetation that are associated with the archaeological contexts that we study. Lipid biomarker analysis started with petroleum research. Geobiologists back in the 1960s, were interested in figuring out the biological origin of petroleum. And around the same time, chemists figured out ways to separate different compounds and developed the technology to do it. George Eglinton, one of the pioneers in the field, investigated succulents native to the Canary Islands and managed to extract their leaf waxes, consolidating the technique of lipid extraction and analysis. Afterwards, the technique became widely used for the reconstruction of past climates. And later on, several researchers, for example, Condamine and colleagues, applied it to archaeology by identifying wine residues in Roman amphorae. And Evershed and colleagues identified some of the products used in Egyptian mummy embalming. Nowadays, we extract lipid residues directly from archaeological sediment. This can be done with very ancient sediments, even with Paleolithic sediments. So how does it work? First, lipid compounds are extracted from archaeological sediment or archaeological materials like bones, stones, ceramics, and other materials. And then the chemical structure of the molecules and their isotopes. And then with a lipid extract, a solution that is injected into different instruments, the chemical structure of the lipid molecules and their isotopes is investigated in search of lipid biomarkers, fingerprints that we know from the literature and from reference collection. How do we collect lipid samples from an archaeological site? First of all, it is very important to collect the sediment or the material with sterilized metal tools. The tools need to be metallic. They cannot be plastic, as plastic is organic. And they must be collected with gloves in order to avoid contamination of organic substance from ourselves. The samples are collected into aluminum foil, glass, or another plastic-free container, and they are stored at low temperatures, ideally in a freezer at minus 20 degrees Celsius. They have to remain there until they are processed at the lab to avoid bacterial activity. How do we extract lipids from archaeological materials or from archaeological sediments? First, we must understand that lipids are normally divided into 5 different groups according to how repellent they are from water, how hydrophobic they are. These groups are the hydrocarbons or alkanes, the aromatics, the ketones, the alcohols, and the fatty acids. In order to separate lipids into these 5 groups, first, we extract what we call the total lipid extract from the sediment or from the material. This is simply a mix of the total lipid content in the sample. Afterwards, we use different combinations of chemical solvents to separate this TLE or total lipid extract into the different fractions, because each of these lipid fractions contains different biomarkers of archaeological interest. Finally, these extracts are injected into a GCMS instrument, a gas chromatography mass spectrometer. And this instrument allows us to identify different compounds. We can also inject the sample into a GC-IRMS, gas chromatography isotopic ratio mass spectrometer, which measures the ratios of different isotopes in the lipid compounds. In the chromatographer, the lipid mixture is pushed by helium through a coiled hollow glass capillary that is several meters long and very, very thin, only a few tenths of a millimeter in diameter. And all of this sits inside an oven at variable high temperatures that enhance the movement of the molecules. Light lipid molecules get to the end faster, and heavy ones take longer. In the end, we obtain a graph called a chromatogram. And these graphs show peaks that represent different molecules ordered by their weight, with the light ones at the left and the heavy ones on the right. Another part of the instrument is the mass spectrometer, or MS. This part is where the specific masses of the molecules are measured. And this allows us to identify the molecules using a standard library of lipid compounds, and later to quantify their concentrations.
This is a typical mass spectrum. What program does is to compare our spectra with the ones in the library. The system provides a potential identification, and with these numbers here, you can evaluate the chances that the identification is correct. The analyst checks these results according to her knowledge.
There are some notable examples of archaeological studies that have used lipid biomarker analysis. For example, one is the case of a series of sites around the Baltic Sea that date to around 6,000 years ago, and which have puzzled archaeologists for a long time. Because at those sites, archaeologists found ceramic vessels. At a time before the arrival of agriculture, sedentism, and animal domestication in that region. How is it possible that there was pottery before the Neolithic Revolution? Ceramic is a material normally associated with agricultural societies. But these are nomadic hunter-gatherers living several thousand years before the arrival of farming. Well, it was a lipid biomarker study that revealed that these ceramic vessels contain a diversity of products including aquatic, animal fat, and even dairy residues. What were these hunter-gatherers eating? They were consuming products that are typical of agricultural society. This was a very unexpected finding. Another example is the case of lipid biomarker analysis on sediment from Paleolithic fireplaces. This study was done using samples of sediment from Paleolithic sites and also from experimental fires that were made as control groups. The results provided evidence of animal bone burning, a group of lipid molecules called ketones that form during the burning of fatty acids, which bones have. This finding is very important because many times Paleolithic fires do not preserve well. They're simply not there. And finding hard evidence of molecular lipid residues of burning is proof that human fire was there. This study is a good example of how the invisible archaeological record can be studied. In this case, evidence of bygone fire.
How much sediment should we collect? It's dark and it looks like there could have been a fire over here. But also, there's volcanic sand and not much clay. It could be very inorganic with a very small amount of lipids.
Yeah, you're right. The usual amount is around 5 grams, which is like 1 teaspoon. But I've seen studies in which they had to collect even 20 grams to get a good lipid signal. Also, let's not forget to collect a control sample nearby to compare your possible fire with the typical sediment of the site. I mean, imagine if we find a plant biomarker, could have been from the plants using the fire or from the natural vegetation at the site, and we should control that.
Yes, and a magic sample. We need to collect a much larger sample, remember? From anywhere in the site. This will be used to know how lipids behave in this type of sediment, and it's minimum 15 grams.
Really, that much?
Yeah, some kind of sediment trap lipids. And we need to know how much lipids are being trapped by our sediment, and then calibrate our results.
Okay, we have a lot of work to do.
Yeah.
Okay, I am weighing my sediment samples now. Then I need to mix them with cycloromethane and methanol, shake them in an ultrasound bath, centrifuge them, filter them, and evaporate them. How long will the entire process take per sample? Can I do one sample in a single day? And can I stop at different times along the process?
Well, that depends on what extraction method you use. And of course, that depends on what you're interested in. Some methods are very fast, like the quenchers, which takes half an hour. But it is only for extracting alkanes. Some lipids are not in their free form and need to be chemically transformed. What we can do is to add a salt to create a soap, so that we liberate the compound that we want to analyze, and this process is called saponification. Or, if you are interested in alcohols like sterols and estanols, they are also in their free form and need to be chemically transformed. This process is called derivatization. Each of these procedures are slow and they may take an entire day.
Okay, what if I want to process my samples to obtain the total lipid extract and then separate all the lipid fraction for later?
Well, in that case, the entire process takes between 4 and 8 hours, depending on the level of expertise of the analyst and on the type of sample. Some samples take longer because they are very complex, and it takes long times to filtrate them.
Okay, can I stop at different times along the process?
Yeah, you can, but I advise you to process between 4 and 6 samples, not more. With that number, you can take breaks in between the different steps of the process.
As we have seen, lipid biomarker analysis is a cutting-edge technique that is contributing valuable information to archaeological research. We can finally access the human organic world in the past. Young archaeologists, historians, biologists, and geologists are getting the necessary skills to learn this technique, and without a doubt, it will soon be an essential technique in archaeology.